and diversity of microbial life. I would say please watch the review video. It's longer and it's a little bit more in detail than we can go right now because we don't have that much time. We only have 10 minutes left. So I'm gonna try to do whatever I can in those 10 minutes. But chapter 15 and 16 review video, I have already posted that. I'm interested in knowing, so before I get started with this. Chapter 15. Life arose on Earth around 4 billion years ago. There might be a question about that. So around 4 billion years ago. And the way we are able to know that, and the way we are able to say this is how it happened, is because of two things. One, Stanley Miller, another name you need to know. Stanley Miller experiment, known as the Miller experiment. That's another name you guys need to know. If you're making a list, put that on your list. Can somebody tell me what Miller experiment showed? Can somebody tell me what Miller experiment showed?
Isn't it the infancy of RNA?
So he took some inorganic molecules. He left the experiment run. So he created similar environment as an early Earth for a volcano.
Oh, amino acids, yeah.
Yeah, he put some fire there for a volcano. So water started boiling. For lightning, he put some electrodes, and then he put inorganic molecules here. And then he let it run for a few weeks. And then in the end, those inorganic molecules turned into organic compound, specifically amino acids. And that's the first theory we have that we base, we say that this is how life came around on Earth. So that to answer the question, how did life arise on Earth? This is one theory we base our base that on. And then later, NASA was able to find these inorganic molecules on the meteorites. So now they have more evidence, but this is the first one that kind of showed that. So that's one thing. Second thing was how genetics came around. Genetics came around with the use of clay. So if you have some A's and G's, they get stuck on clay. And if you have the complementary base pairs nearby, they're going to attach. So clay might have catalyzed a reaction. So those are two things that we base how the life on Earth came around. Once that happened, and RNA came around first because it's simpler. DNA is more stable, so it got evolved over time, and then DNA came around. So sometimes some of the organisms still have RNA, and then some are both. So this is your first cell, protocell, prototype. So you have a simple cell, you have the lipid membrane, and then you have some amino acids or proteins, and then some DNA. So that's your first cell, very simple. Now, once you have your first cell, now over time, it's gonna get more complex, and it's gonna be becoming a prokaryote, then a eukaryote. So we have different groups under prokaryotes and then different groups under eukaryote. So in that video that I did, I basically wrote everything out. I redrew this and then kind of wrote what you need to know for each one of those. So I think that would be helpful. I can't do that right now because I don't have that much time today. Prokaryotes are single cell organisms. Eukaryotes are multicellular. Under eukaryotes, animals, we are in this phylum, Animalia, fungi, plants, and protista. So there are a few things I want you to do for prokaryotes. I want you to create flashcards for these 3 slides. For prokaryotes, you have bacteria and archaea. You need to know they have no nucleus. You need to know the size. I will ask you the size. And then some other characteristics. So try to make a flashcard for this, and that some can fix nitrogen and grow at temperatures above 80 degrees. You need to know what the cell wall is made of, peptidoglycan. And they can do photosynthesis. They cannot generate methane, and they are sensitive to streptomycin. Know the name of this antibiotic, streptomycin. And there are no introns. Hopefully you guys know what introns are. They are kind of genetic material that doesn't code for anything. Then this versus archaea. Archaea are the ones that are found in acidic environments, acidophiles. So they're known as acidophiles if they live in acidic environment. They live on high salt environments. They live in extreme environments. Not all of them though, some of them. And they can generate methane. That's why I showed you that if there's standing water, there's no oxygen left in there, and these are going to be found there, they're going to make methane. And sometimes if there's lightning, it can put that on fire. They're insensitive to streptomycin. They can contain introns. Again, the best way for me to tell you how to memorize this is making a flashcard. Make 3 flashcards for these 3 slides. Then you're gonna, again, you can create a table for this. This is gonna go over what prokaryotes has. It has a nucleoid. It does look like a nucleus, but it's not nucleus. It has plasmids, pilus. Pilus helps it to attach to surfaces. Also helps, there's something called sex pilus or sex pili that's going to help it exchange genes. Flagellum is a tail, cell membrane, and cell wall. What is cell wall made of for bacteria?
Peptidoglycan.
Peptidoglycan. Peptido means the protein, glycan is sugar, protein and sugar. Plasmids are circles of DNA that can replicate themselves. You already should know this definition from chapter 11. Cell wall is going to give it more protection. 3 shapes of bacteria. Again, they're multiple, but I only want you to know 3. Circles, spheres, coccus or cocci, bacillus, rods, and spiral. It looks like a spiral or corkscrew. On the play pause video, I gave you a question that's going to have you, answer all 3 types of 3 shapes of bacteria. They can make endospores. That means they take all their genetic material, put it in this endospore because they're dying. And then when the temperature is right, they can start, dividing again and grow again. And I gave you example of Clostridium botulism. If you have a canned food and there's no oxygen in there, and that can starts to look like this. Like this, it starts to look like this. Please do not eat that. That means there is bacteria in there that's making that gas and causing the can to look like this. So that's Clostridium botulism. Is a type of bacteria that can make endospores. These endospores can survive very, very harsh conditions. Another flashcard for this. Know what autotrophs are? Which ones are autotrophs? Heterotrophs, protista. Some of the protista is heterotroph, some of the protista is autotroph and phototroph. Chemotrophs, obligate aerobes, and anaerobes. This would be an anaerobe because it can live without oxygen. Then we start dividing them into different groups. So for bacteria, we have proteobacteria, cyanobacteria. Under proteobacteria, we have E. coli and Salmonella. It's better if you draw it out. So you can write bacteria, then under bacteria, you can write proteobacteria. Under proteobacteria, you can write E. coli and Salmonella. Another group of bacteria, you can write cyanobacteria. Again, this is what I'm talking about. I drew it out for you. So please watch that if you're having a hard time with the groupings. Archaea, they're found in extreme environments. They are extremophiles. So environments that are salty, acidic, hot. Some live in moderate environments. Bacteria and archaea, they're essential to life. They do nitrogen fixation. Otherwise, all the nitrogen in the environment would be locked up. We wouldn't be able to use it. So that's why we need bacteria and archaea for that. And then not all the bacteria is harmful. Some is harmless. So if you don't have bacteria, and if the bacteria gets in a space where it's not supposed to be, then it's going to cause disease. So that's harmful. If it's, not in a place where it's not supposed to be, it's not going to cause disease. And then bacteria and archaea, So for the infections, you can take antibiotics and your doctor's gonna say finish the whole course. So if it's a broad spectrum, antibiotics means it's going to target more than one type of bacteria. It can also kill some good bacteria in your stomach. That's why sometimes you get diarrhea when you're taking some of the antibiotics. And then it takes some time for you to go back to normal. So the good bacteria that also got killed with the broad spectrum antibiotics, that's going to have to grow back. You're going to have to eat some yogurt or some fermented tea or something like that to just, kind of regrow that bacteria there, or repopulate that space. If it's narrow spectrum, it's only going to focus on one type of bacteria, then it's not going to lead to secondary infections. Some of the bacteria is used to make cheese, make yogurt, make sourdough bread. Sometimes people use yeast. So if you're interested in actually looking at the process, I know Bernice was, you can kind of go on YouTube and watch that. It's also used to make insulin. So those are some good uses of bacteria. Then we have eukarya. We are out of time. Do you guys want me to continue going? So I'm gonna finish this. If you can stick around, do that. If you can't, then just come back and watch the recording, okay? So for eukarya, we have 4 groups: Protista, fungi, plants, and animals. I'm gonna do Protista, fungi for this chapter. Plants is a different chapter. So very important. How did the eukaryotes came around? I told you guys we had a protocell, very simple cell, just the plasma lipid, the fat around it, and then genetic material inside and some amino acids or proteins. Then this membrane that was on outside, it started folding in. It started to look like this. This gave it more surface area. Now it had more surface area for internal structures. And then the way some of the organelles came around, they started eating small organisms that became their organelles later. This theory, very important. There will be few questions on this. It's known as endosymbiotic theory. And the 2 organelles I'm talking about here is mitochondria. It was its own organism, but it was really good at making energy. So this cell, bigger cell, ate it, and it said, I'll give you space to live. You give me energy in return. So they started living together in a symbiotic relationship. And now this bacteria cannot, or this organism, even we have mitochondria, cannot live without mitochondria. It's dependent on mitochondria. Another bacteria, photosynthetic, is chloroplast. So it's eaten up by plants. So bigger organism again engulfed this chloroplast and became an organelle. This is also making energy with sunlight. So that's how some of the organelles came around. So I would say, what is endosymbiotic theory? You're gonna say that it's when free living bacteria was engulfed by other microbes. And I might say which 2 organelles came around because of endosymbiosis. You're gonna say chloroplast and mitochondria. And then sometimes there is a secondary endosymbiosis. That's when another even bigger organisms ate that organism that had already eaten the mitochondria. So that's secondary endosymbiosis. Then the bridge between single cell organisms and multicellular organisms is a Volvox. So you have all these are cells there, and these are the reproductive structures. So this is when it became multicellular. Okay, so I'm just gonna summarize this. So for Protista, we have algae, that's photosynthetic. Therefore, they're autotrophs and phototrophs. Slime molds and water molds, they look like fungus, but they're not fungus. Protozoa, they behave like animals, but they're not animals. So in Protista, we're putting everything together that's not animal, that's not plant, that's not fungus. Even though this one behaves like animal, but it's not animal. This one looks like fungus, but it's not fungus. This one looks green like plants, but it's not plant, it's algae. They put these organisms together in Protista based on their DNA. So they look at their DNA and they say they are closely related. They put them in this group called Protista. For algae, do you guys remember which ones we had?
Red,
Yeah.
brown, and green.
Brown, green.
Red, brown,
Red.
and green. Can you give me an example of green algae? You might see it when you go to the beach. Looks like plants. So
Kelp.
Paula?
Kelp.
Kelp. Red algae, they have these photosynthetic pigments that help them do photosynthesis using red and blue light. And then green algae, they are ancestors of land plants. They're similar to land plants. So that's green algae. So here we covered brown algae, kelp, red algae, yeah, red and blue light, and then green algae. They are very diverse, and I want you to know volvox. They are ancestors of land plants. Okay, and then there are two more that do photosynthesis, diatoms and dinoflagellates. Tell me why dinoflagellates are called dinoflagellates.
They have two flagella.
two flagella.
structure.
They have two
Yeah.
flagella, they have two tails. These are the ones that are responsible for red tide. They produce toxins that can kill lots of animals altogether, those toxins when they release these. Diatoms, what do they have in their cell walls?
Silica,
Silica. So that same thing, the mirror is made out of, glass is made out of silica. So diatoms have silica and they are found in moist habitats. So we covered 5 now for algae. Protista, under Protista, we have, actually, let me start again. Eukaryotes, under eukaryotes, we have Protista. Under Protista, we have algae. Under algae, we have brown algae, green algae, and red algae, diatoms, and dinoflagellates. Second group for Protista, we have slime molds and water molds. For water molds, only thing I needed you guys to know, they're decomposers and parasites. For slime molds, they're like transformers. They can exist as single cells or multi-cells. You don't need to know the life cycle, but that's all you need to know. And they're very intelligent at finding food. They're very good at that. So these are slime molds. Third group, protozoa. So that's the one that behaves like animals. So under eukaryote, you have Protista. Under Protista, the third group you have is protozoa. Under protozoa, you have ciliates. Under ciliates, you have paramecium. So that's what I need you to know. So under Protista, third group is protozoa. Under protozoa, you have ciliates. Ciliates, the one that have these hair-like structures. And this is called paramecium. So I gave you the name paramecium. That's another one I want you to know. So you can expect similar questions like this. So make sure you kind of know the characteristics for each. Then we have fungus. Very important. So fungus, they're heterotrophic. Here, protozoa, they're heterotrophic. If it's something that's going to do photosynthesis, you already know it's a phototroph and autotroph. But if it's feeding on others, it's a heterotroph. Like this paramecium, you find it in pond water. It's kind of swimming around. It's using the cilia and it's grabbing onto things and eating those. So it's a heterotroph. And fungi are heterotrophic, and they also decompose. So 3 keywords I want you to know: mycelium, fruiting body, and hyphae. There'll be 3 different questions on all these 3 parts. Do not get them confused. This is the fungus. This is a reproductive structure it makes when it wants to reproduce because it's moist, it rained, there's nutrients or something like that. When you zoom in on mycelium, the fungus, you have these structures called hyphae, multiple, hypha, singular. You don't need to know the different names, but you do need to know they make spores. And you also need to know what dikaryotic is.
Professor, which one did you say was the reproductive structure?
The fruiting body.
Okay.
So know that they make spores. You don't need to know this life cycle, but you do need to know what dikaryotic means. I just showed you this hyphae. Let's zoom in on the hyphae. This is what it looks like. There is a nucleus inside. There's one nucleus inside this cell, another one here. These are haploid. So haploid cells, one nucleus. These hyphae fuse together. Now this cell has two nuclei. This is very unique for fungus. Only fungus does this. So that's why I want you to know this. Dikaryotic. Di means two nuclei. Only this stage is called dikaryotic, two haploid nuclei. Once these nuclei fuse together, now they're diploid and it's one nucleus. This is not dikaryotic anymore because you don't have two nuclei, they're fused together. Only this stage is called dikaryotic, and this happens through meiosis in fungus. So again, you don't need to know this life cycle, but you do need to know dikaryotic. Fungus, they are used as food, as medicine, and they can also form positive symbiotic mutualistic relationships with plants. They can help plants grow better because they are giving them water and then minerals. Plants in return are giving them sugars. That's called mycorrhizae fungi. So know this term, mycorrhizae fungi is the one that's in a symbiotic relationship with the plants. It's getting something in return and it's giving. Last term, lichens. Lichens are also fungi with green algae or cyanobacteria. Green algae is Protista. We just covered it. So lichen would be some type of algae plus cyanobacteria. Cyanobacteria is a type of algae. It's a very general term that's living with, so algae and fungi are living together here. So algae and fungi is making a lichen. In other words, fungi and Protista can also make a lichen because green algae is type of Protista. So kind of, think it through. 3 diseases for fungus you guys have to know. Cordyceps, that's the zombie ants. Fungus starts to grow out of their brains, takes over. Powdery mildew, you've probably seen this on plants. It's powdery stuff that's coming from fungi. And then nose syndrome for the bats can happen in bats. It's also from fungus. That's it for chapter 15. There was a lot in this, but again, if you watch that video, it's really going to help you with this chapter.